Good afternoon, ladies and gentlemen. It's a pleasure to be here today to talk about our technology, give you an overview. I want to thank the Alliance for having us, and we've been here for about 5 years now where we presented, both Bob and I, and I'm happy to announce that we can finally change our name from preclinical to becoming a clinical stage company. And we are a regenerative clinical stage medicine company developing regenerative therapies that alleviate pain and restore function in patients with degenerative diseases of the spine. And to give you a little bit of history on our company, we were founded in 2007. We are now located in Salt Lake City, Utah since 2012. We are privately held. We have 67 accredited investor and one VC who just recently joined our company. We have an outstanding advisory team panel that not only consists of renowned neurosurgeons and spine specialists, but also excellent legal, accounting, and other ancillary support systems to support our endeavor. We have a tremendous scientific and management team, and I want to talk about that scientific team briefly because they're an incredible group of people. Laura Silverman, Galina Delatova, and Terry Tandeschi started with us really in 2012 as Bob Wintelak drove our preclinical development. And Laura came from the University of Pennsylvania, and she's done just an extraordinary job of working with Galina and Terry Tandeschi and driving our U.S. initiative. Isaac Erickson came from Biotime back to his home state of Utah in 2014, and he has been instrumental in our Japanese initiative. Our management team consists of Bob Wintelak, Dr. Kevin Foley, and myself. Bob Wintelak is an industry biomedical and spine veteran. In 2005, he was the president of Osteotech as a team that rebuilt Osteotech, which was purchased by Medtronic in 2010. Dr. Kevin Foley is considered by many the father of minimally invasive spine fusion. He has 110 issued patents, of which about 50 of those are licensed by Medtronic that offer up their minimally invasive spine technology. He is a tremendous asset to our team and has helped us drive to our clinical state that we're in today. So we're really trying to address low back pain, and it is a global health problem. It affects a quarter of the global population. It imposes a major financial burden on the healthcare system at greater than $100 billion spend in the U.S. alone. It is the leading cause of disability worldwide. It obviously leads to missed work, lost wages, addiction to pain medication, including opioids, which, as many of you know, I'm sure, if you read the newspapers, is a serious public issue, health issue in our country, and we hope we can help in alleviating that and maybe answering part of that problem. Low back pain in this area, it really is void of curative treatment options. And this degenerative disease of the spine is a major contributor to low back pain, and it is characterized by inflammation and breakdown of extracellular matrix within the spinal disc tissue between the vertebral bodies. And current treatment options really are limited to pain management, physiotherapy, and then in the end, surgery as the degenerative cascade and that rock rolls down the hill. And it really is progressive and cyclical in nature, which really starts out with injury, possibly genetics, and especially the population aging and becoming more active. And you get into this mild stage, which progresses to a moderate stage. And in the mild stage, you're trying to do pain meds, NSAIDs, physical therapy. It possibly leads to steroid injections, ablation, chiropractic, and alternative care. And then when you move to the moderate stage, you really are in some ways just buying time or escalating some of the mild stages of pain reduction. And so we believe that if you're going to try to solve this problem, you do it in this mild to moderate stage, because in the severe stage, you really are probably ready for surgical intervention, because the pain is so great. And you can see here, this is a 2-level fusion using plates, rods, and screws to lock down those vertebral bodies. And what happens is that the disc level above and below this fusion, usually over a 5 to 8 year period, results in adjacent level disease. So this is our approach. We are harnessing the restorative potential of cells native to the intervertebral disc to develop what we hope will be a profound therapeutic option for millions of patients suffering from the debilitating effects of back pain. And these cells are specifically engineered to uniquely address the complex environment of the degenerated disc. We use these therapeutic progenitor cells that originate from the exact cell type that are intended to be regenerated. These cells possess key regenerative properties that are very similar to MSCs, i.e. the anti-inflammatory effect, but they also produce the same extracellular matrix components that comprise the nucleus pulposus of the disc. So we offer a critical advantage over common cell-based approaches, and we are now reproducibly manufacturing these cells in a CGMP process, and they are protected. The cell process is protected by 24 issued patents and 15 others that are pending. If you look at the discogenic cells compared to generic stem cells, we believe that the introducing of therapeutic cells that originate from the exact cell type that they are intended to regenerate offers a critical advantage over other cell-based approaches that are attempting to use cells derived from either adipose fat or bone marrow aspirate that must first adapt to the unique environment of the disc before they can attempt to have any effect on it. And our cells are done almost exactly the opposite way. They are driven to their progenitor state. They are from disc, for disc, to help disc, and when they hit the disc in their injected state, they go right to work, as opposed to interrogating and trying to become a cell to help that state. Our first product candidate is IDCT for degenerative disc disease, and IDCT stands for injectable discogenic cell therapy. It is a homologous allogeneic injectable cell therapy for the treatment of patients with this mild to moderate degenerative disc disease. It is cost-effective, non-surgical, off-the-shelf. It is comprised of these discogenic cells in a viscous delivery delivery vehicle. And we are currently underway clinically in the U.S. and Japan to evaluate the efficacy. The treatment is a percutaneous injection in a treatment room with a C-arm, non-surgically by syringe, and our post-treatment hypothesis is to reduce pain and normalize disc tissue. As you can see from our IND-enabling GLP study of 45 rabbits, we induced a degenerative model where we drove these disc heights down to about 60 to 50% and injected. As you can see from the results, we had restoration of disc height and normalization of tissue architecture not seen in the controls. We had no toxic or safety concerns, and as you can see, we had some very nice disc height restoration and histological normalization of tissue. This was our IND study, and this was preceded by another 14 animal studies we've done over the last 5 years. Another study that I'll show you was another IND-enabling GLP tumor study with 30 athymic mouse in their subcutaneous patch. It was really interesting in the fact that we were using this study for tumor genicity, and we found that there were no safety concerns or tumor formation, but more importantly, at the injection site, we revealed that there was formation of disc-like tissue. And you can see this from the histology that not only did we show a lot of proteoglycan production, but we did so, really what was aligned collagen fibers that looks very much like a disc. So in our clinical pipeline, currently we have been approved to open a 60-patient, double-blind, placebo, saline-controlled, multi-center trial here in the U.S. We hope to have patient enrollment in Q4 of this year. We are preparing to submit our CTN to the Japanese PMDA, and we believe that there will be follow-on indications around cervical DDD, adjacent level, and other herniated disc opportunities. We are at a CGMP manufacturing scale-up, and that is now underway as we transition from preclinical to late clinical and commercial phase. We are doing this ourselves, and as part of the transition, we are evaluating various manufacturing technologies that will facilitate the optimization of our single-lot manufacturing process, inclusive of maximizing dose output and reducing cost. And this reduction of cost, we believe, is very important to us, and I'll show you why in just a second. Another important factor is in obtaining our phase 3 CGMP process is to ensure that we are meeting all global compliance requirements. And this includes removing certain serums, closing the system, adding additional and other process improvements. So in 2013, we really looked at the prioritization of global markets, and obviously we came up with the US and Japan, and we were really excited by the accelerated pathway in Japan in 2014. And so we have gone after both of these markets, and as I said, we're very close to clinical trials in both those countries. And really it is our hope from our scale-up to commercial GMP manufacturing process that we could do global distribution all over the world from our cell facility in Salt Lake City. If you look at the market size, it is huge. Projected to be $15.2 billion in the US by 2022, and projected internationally to be $30.4 billion for a total disc repair market of about $45 billion. Through our technology, we really believe we can disrupt the treatment continuum. Current degenerative treatment pathway example can go from 2 to 5 years, resulting in a surgery, and that pathway cost exceeds $115,000. We really believe after 3 months of conservative care, you could do a single dose of our product for about $15,000. And if we do restore function, you can see it has a dramatic reduction in cost to the entire healthcare system, not only in outlay of dollar cost, but time, and it definitely increased to the patient's quality of life. So far in our fundraising, we just completed a Series B round in August. It was jointly led by Mitsubishi Capital of Japan, our board of directors, and existing long-term investors. We are currently raising our Series C round. So in summary, we now are a clinical stage regenerative medicine company focused on treating degenerative diseases of the spine. We have a very experienced management team with strong scientific expertise addressing a significant unmet medical need that really is void, that really lacks curative treatment options. Our first product candidate is cost-effective, non-surgical, off-the-shelf combination cell therapy to treat degenerative disc disease, and we really believe these proprietary DiscGenics cells originate from the exact cell type they are intended to regenerate in a homologous allogeneic application. So we believe it offers critical advantages over bone marrow or adipose fat cell-based approaches, and we have strong preclinical data that include disc height improvement. We are initiating clinical programs in both the US and Japan, and we are manufacturing scale-up for phase 3 commercial production, which is now underway. We really believe that we have the opportunity to help many, many millions of people that are debilitated by this pain, and we're looking forward to the outcomes of our clinical trials. Thank you very much. Any questions? Great.
There's one.
Sorry.
Oh, sorry, wait.
Your source of cells, are they cadaveric? And if so, what's the age range?
So our cells in our tissue is in organ procurement. So when an organ donor, beating heart death, We go in, or the organ procurement team goes in, and when they're taking heart, lungs, liver, they take 6 to 10 levels of disc tissue. And that is the single lot product that they deliver to us, and then we run it through our culture system, which is a 2-step process, and it takes approximately 7 weeks.
Thank you.
Yeah, my pleasure. We think that obviously is very scalable. We believe we can generate multi-thousands of doses, which is why we believe we can deliver at this kind of cost relative to the cost of that long-term existing treatment continuum, which would possibly include a surgery that is well above $115,000. And we also think if we can interdict early, we also think this has a significant impact on this whole notion of opioid addiction in this country, and really some of that around the world. Anything else? Thank you very much. I appreciate it.